Just make sure everything is perfect from my side as well as from yours. Am I audible to you? Am I audible to you? Am I audible to you? Confirm me everyone that am I audible to you? Am I audible to you? Just confirm me, am I audible? Everything is perfect. Okay, fine. See, if you find any difficulties, just intimate me very soon. Okay, fine. Okay. In today's class, we are going to deal with the carbohydrate metabolism, most specifically cellular respiration. Okay, so without any delay, we'll start at today's class. See, carbohydrates. There will be different types of carbohydrates. We can classify based on their number of carbon. Okay. And that will be a monosaccharide, disaccharide, and polysaccharide. See, based on the hydrolysis, when we hydrolysis a particular sugar, if we get only one saccharide, okay, if we can't hydrolysis, then it will be called as the monosaccharide. If on the hydrolysis we can get two sugar, that will be a disaccharide. If we get many monosaccharide as a result of hydrolysis, then we are calling them as the polysaccharides. Okay, you should know that glucose and fructose, that will be the monosaccharide. Sucrose and lactose will be the disaccharide. Why? Because on hydrolysis, sugar can produce glucose plus fructose. Okay, on hydrolysis, lactose produce glucose plus galactose. On hydrolysis, this maltose gives rise to glucose plus. Okay, see, I hope everything is perfect, right? Audio, something I find any trouble. Okay, fine. Okay, where it's, yes. When we hydrolysis this polysaccharide, we can produce many monosaccharides. Okay, this is what types of carbohydrate is. And how can we define this particular carbohydrates? See, carbohydrates or the organic molecules, it composed of carbon, hydrogen, and oxygen. That will be in the ratio of 1:2:1, which means what? CN H2O of N. Okay, this is what a formula of the carbohydrates. The carbon will be in the ratio of 1 and hydrogen will be in the 2 and oxygen will be in the 1. And this carbohydrate will be act as the primary energy source. Okay, now the metabolism. How the carbohydrate is getting metabolized in our body, that will be glycolysis, citric acid cycle. It is also called as the TCA cycle or the Krebs cycle, and oxidative phosphorylation. Okay, this is how the carbohydrate is getting metabolized within the body. Now, what is glycolysis? In glycolysis, we are breaking down this glucose into the pyruvate. Okay, as a result, we are getting ATP and NADH. And like in the same way, in the Krebs cycle, here the pyruvate is getting breaking down. As a result, they are producing the ATP, NADH, and FADH2. And in case of oxidative phosphorylation, here the ATP synthesis using NADH and FADH2. Based on there will be an involvement of several electron carriers, and through the carriers, we are producing the ATP with the use of, with making use of this NADH and FADH2. Now, what is the purpose of energy? Purpose of energy in the sense means how the carbohydrate is getting converted into an energy and what is their role. And this carbohydrates will be considered the primary source of the energy for cells. Okay, and most particularly, this glucose will be the energy fuel of the cell. Okay, especially in the brain and muscles. See, even if we work physically, if we work mentally, which sucks more energy means when we work mentally. Okay, it results in draining of energy. Okay, so which means what? The brain needs most of energy, right? Now, what will be the chemistry of the carbohydrates? Here we know that it composed of carbon, hydrogen, and oxygen atom, and this will be their general molecular formula, CnH2O of N. And what is their function? We know that energy protection, that will be their ultimate goal, and structural support. For example, in plants, cellulose is exclusively, it is carbohydrates, and cell signaling, and it will be glycoproteins and glycolipids. Of course, it will be the composition, it will be the combination of proteins and lipids. There will be the involvement of carbohydrates also. And how it is getting regulated in health? Critical for maintaining the blood glucose levels within a narrow range. This regulation leads to, if there will be imbalance in the carbohydrate maintenance, it results in the diabetes mellitus and some of the metabolic syndrome. Okay, and how the carbohydrates is important in the diet. Okay, see, it will be found in the grains, fruits, vegetables, and some of the dairy products. Right now, here, what is cellular respiration? See, you should not think that cellular respiration is only glycolysis and the Krebs cycle. Okay, but this cellular respiration involves the four stage. Okay, first thing, in this process, the glucose is getting oxidized into the carbohydrate and oxygen is reduced into the water. See, here the glucose is oxidized into the carbon dioxide and oxygen is reduced into the water. See, you should know what is oxidation and what is reduction. Here, the oxygen is getting accepted. Here, hydrogen is getting accepted. Here, the electrons will be lost here in oxidation. In reduction, there will be a gaining of electrons. Okay, so you should know the difference between the oxidation reaction and the reduction reaction. Okay, see, if it is NAD+, it is in the oxidized state. And if it is in the NADH2, this will be in the reduction state. Okay, there will be an acceptance of hydrogen. Here there will be the removal of hydrogen. So this will be the difference between oxidation and reduction. Now, see, as a whole, in cellular respiration, they produce 38 ATP molecules. Okay, 38 ATP molecules as a net product. Okay, and 36 will be their gross ATP production. Okay, and this cellular respiration involves 4 stages. You should not think this glycolysis alone will produce a 38 ATP molecules. That is wrong. But instead, it includes 4 stages. One is glycolysis, and second one is pyruvate decarboxylation, and the third one will be the Krebs cycle, and the fourth one will be the electron transport system and oxidative phosphorylation. Okay, when it completes these 4 stages, we are producing this 38 ATP molecule. Now, what is glycolysis? Here, there will be an oxidation of glucose molecule. Okay, by oxidizing this glucose molecule, we are getting 2 molecules of pyruvate. Okay, and where is this process of glycolysis is taking place? It is in the cytoplasm or cytosol. Now, the second, after the pyruvate is getting formed, this pyruvate has to be converted into acetyl-CoA. Okay, and that process is called as the pyruvate decarboxylation. Now, see, we are getting the pyruvate from the glycolysis, and this pyruvate now it is enter into the mitochondrial matrix. There, it is undergoing an oxidative decarboxylation, which means what? Removal of carbon dioxide. Okay, as a result of removing this carbon dioxide, they are producing two molecules of acetyl-CoA, and this reaction is getting catalyzed by, or this reaction is getting enhanced by the enzyme called the pyruvate dehydrogenase enzyme. Okay, now the third step will be the citric acid cycle. Okay, see, it is a common pathway. The first statement alone can be raised into a question. You should know that whatever be the cycle is, whether it will be the carbohydrate, whether it will be the protein, whether it will be the lipid, everything will be connecting through this particular cycle. Okay, it is a common pathway for all these carbohydrates, proteins, and lipids and all. See, here they are getting metabolized to the acetyl-CoA enzyme, that is acetyl coenzyme A, or other intermediates of the cycle. Okay, see here this acetyl-CoA enters into the citric acid cycle. Okay, now here the glucose is getting fully oxidized. Now this acetyl-CoA is combined with the OAA, oxaloacetic acid. How many carbon is present in the oxaloacetic acid? That will be 4 carbon compound. Now acetyl-CoA plus this oxaloacetic acid together, combined together to form the citric acid. Okay, citric acid contains a 6 carbon molecule. Okay, now in this process 2 molecules of carbon dioxide is getting released. Now this oxaloacetic acid will be recycled and the cycle continues. Okay, now here the energy is stored in ATP and high energy compounds like NADH and FADH. Everything will be produced in case of citric acid cycle. Now electron transport system and oxidative phosphorylation. Here, the ATP is generated when the electrons are transferred. See, whenever the electrons are transferred through the electron carriers, there will be a lot of carriers will be there. This electron carriers will be transferring their electrons from one carrier to the another carrier. When it is transferring the electrons, the energy is getting released, and that energy released will be used for the synthesis of ATP. Okay, this is what happening in the electron transport system. Okay, now see here the electrons are transferred from the energy rich molecules like the NADH and FADH. See, approximately 1 NADH is equal to 3 ATP, or we can say 2.5 ATP, and in FADH 1 is equal to 2 ATP. Okay, or 1.5 ATP. So this is how this will be the energy rich molecule, and this will be producing the glycolysis, citric acid cycle, and the fatty oxidation to molecular oxygen by the series of electron carriers. Since the electrons are transferring through this particular electron carriers, when the electrons are transferred from one carrier to another carrier, some sort of energy is getting released, and the energy released here will be used in the synthesis of ATP. This is how the ATP will getting synthesized. Now, here the oxygen is getting reduced into water. Here, what is reduction? Obviously, there will be the acceptance of hydrogen molecule. Okay, by accepting the hydrogen molecule, this oxygen is getting reduced into the water. Now, where it is takes place? It takes place in the inner membrane of the mitochondria. See, the location is very, very, very important, where all these steps, 4 stages are taking place under the cellular respiration. So that's all about the 4 stages of the cellular respiration. Now coming to the glycolysis, the most important cycle. See, initially the glucose will be converted into the glucose 6-phosphate, and this reaction is getting catalyzed by an enzyme called the hexokinase. Whenever the kinase enzyme, the enzyme will be having a suffix called the kinase, definitely the enzyme has a function. They are going to add in the phosphate group. Okay, whenever the enzyme the name of an enzyme ends with a kinase, their duty is to perform the addition of the phosphate group. Okay, that is why here with the use of the ATP, the glucose is converted into the glucose 6-phosphate. Now, where the phosphate group is added in this particular glucose means at the 6th place. That is why the name is glucose 6-phosphate. Okay, now this glucose 6-phosphate is getting converted into the fructose 6-phosphate. Okay, here the chemical formula doesn't change. Glucose is also C6H12O6 and fructose is also having a C6H12O6. But what is the enzyme is converting this glucose 6-phosphate to the fructose 6-phosphate will be the phosphoglucoisomerase. Okay, isomerase enzyme. Whenever the enzyme name of an enzyme ends with an isomerase, definitely the reactant, the isomer of the reactant will be the product. Okay, see, the number of carbon doesn't change. This is what a function of the isomerase enzyme. Now, the fructose 6-phosphate is getting converted into the fructose 1,6-bisphosphate. How is that possible? Again, the enzyme phosphofructokinase. Once again, if the enzyme kinase will be there, definitely there will be the addition of phosphate group. And where is the phosphate group is getting added? In the first place of the fructose. That is why the name fructose 1,6-bisphosphate. Okay, now see here with the enzyme aldolase is getting converted into the 2-glyceraldehyde 3-phosphate and dihydroxyacetone phosphate on the other side, DHAP, dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate, and these dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate will be isomeric form with each other. Okay, and that will be in the equilibrium state, just like glucose 6-phosphate and the fructose 6-phosphate. Now, this glyceraldehyde 3-phosphate with the use of an enzyme glyceraldehyde 3-phosphate dehydrogenase enzyme. Whenever the name of an enzyme ends with dehydrogenase, there will be the production of the NADH. Okay, with the use of this particular enzyme, the glyceraldehyde 3-phosphate will be converted into the 1,3-bisphosphoglycerate. Okay, now, once again, the enzyme phosphoglycerate kinase. As a result, it will be formed 3-phosphoglycerate. Okay, now, phosphoglycerate mutase, definitely that will be converted into the 2-phosphoglycerate, and after that, with the help of an enzyme enolase, it will be converted into phosphoenol pyruvate, and with the enzyme pyruvate kinase, it will be converted into the pyruvate. And as a result, this pyruvate is getting converted into the 2-acetyl-CoA. This reaction is called as the pyruvate decarboxylation. Okay, this is what a process of glycolysis is. See, here you should know that what will be the net product and what will be the gross product, how many ATP is getting synthesized, how many NADP is getting synthesized, net and gross. Now, and each reactions, okay, reaction is necessary, and which reactant is getting converted into which product, and it is catalyzed by which enzyme. So overall, you have to read all the steps, and also you should focus on which reaction is a reversible reaction and which reaction is irreversible reaction. Okay, all the things is very important. Now, the citric acid cycle. See here the acetyl-CoA. Okay, we are getting glucose is getting converted into the pyruvate, and pyruvate is converted into acetyl-CoA, and now this acetyl-CoA, when combined with the oxaloacetic acid, it will be converted into a citrate with the help of an enzyme citrate synthase. Okay, now by eliminating a water with the use of an aconitase, the citrate is converted into isocitrate. Okay, now this isocitrate with the help of an enzyme isocitrate dehydrogenase, it will be converted into alpha ketoglutarate. In this reaction, the carbon dioxide is getting released and the NADH is produced. Okay, now this alpha ketoglutarate will be converted into succinyl-CoA, and this reaction is getting catalyzed by the alpha ketoglutarate dehydrogenase. Whenever the enzyme ends with the dehydrogenase, definitely there will be the production of NADH. Don't forget this. And in this particular reaction, the carbon dioxide is getting released. Like in the same way, here also the carbon dioxide is getting released. Now this alpha ketoglutarate will be converted into succinyl-CoA, and the succinyl-CoA will be converted into succinate. Here, the removal of CoA group, that is coenzyme A, is getting released by an enzyme called the succinyl-CoA synthetase. Here, the ATP is not getting produced. Instead of that, the GTP is getting produced. Okay, here the GTP is getting produced, not ATP. Okay, so this is where you have to focus on. Now, this succinate is getting converted into fumarate with the help of an enzyme succinic dehydrogenase. Once again, when it will be the dehydrogenase enzyme, they are going to produce either NADH or FADH2. As it is a fumarate, it will be producing the FADH2. Now, this fumarate will be converted into the malate by the enzyme called the fumarase with the addition of the water. Now, this malate is once again converted into oxaloacetate with the help of an enzyme, malate dehydrogenase. And this is a dehydrogenase enzyme. Once again, the NADH is getting produced. This is what a Krebs cycle is. Now, what will be the reactant and what will be the product here? In glycolysis, here the glucose is getting oxidized to two molecules of pyruvate. Okay. Now here, what will be the reactant? What is getting actively involved in this particular cycle? Glucose, ATP and NAD+. As a result, the glucose is getting converted into 2 pyruvate and the 2 ATP is getting used, 4 ATP is getting produced. Okay, as a result, and this 2 ATP is used in the cycle, the net will be the 2 net ATP and this 4 ATP will be the gross ATP produced in the cycle, glycolysis cycle. And how many NADH is getting produced in case of glycolysis? See here, 2 NAD plus is there. It is in the oxidized state. Now this 2 NAD plus is reduced into the NADH. Now, pyruvate decarboxylation. This is called as the link reaction for this particular glycolysis and the Krebs cycle. Now, in the pyruvate decarboxylation, this pyruvate is getting converted into acetyl-CoA and producing the NADH and CO2. Here, what will be the reactant is getting actively involved means pyruvate, CoA and NAD+. This pyruvate is getting converted into acetyl-CoA. Here, as it is a decarboxylation reaction, definitely there will be the release of carbon dioxide. And this NAD plus is getting converted into, is getting reduced into the NADH. Now, in citric acid cycle, this acetyl-CoA is enters into this particular cycle in the redox reaction. What does mean by redox reaction? Reduction reaction is also be there. Oxidation reaction is also there. And reduction and oxidation is occurring simultaneously. It is called as the redox reaction. As a result of this redox reaction, they are producing NADH, FADH2, GTP and carbon dioxide. Here, acetyl-CoA, NAD plus, FAD plus, ADP plus PA. Everything will be converted accordingly. Right? Now, in the electron transport chain in oxidative phosphorylation, here, this NADH and FADH2, from these previous steps, glycolysis, pyruvate decarboxylation, citric acid cycle, everything will be donating the electrons to the electron transport chains. Okay, in this particular chain, these electrons are getting donated. Okay, and this drives the synthesis of ATP. And as I said before, whenever the electrons are getting transferred from one carrier to the another carrier, there will be a release of some sort of energy. And as a result, that energy is used in the production of ATP. That is what the cycle is. Here, this will be your questionable area, which will be the final electron acceptor. That will be the oxygen here. And here, NADH, FADH2, ADP plus PA and oxygen is getting involved. As a result, we are producing the ATP and the water. Okay. Now, one more two cycle is there. One is pentose phosphate pathway and this will be the gluconeogenesis and oxidative decarboxylation. Lot of things has to discuss. So we'll see some of the questions first. Okay. Tomorrow's class will discuss the remaining cycle. Which of the following is a monosaccharide? On hydrolysis of this maltose, what we are going to get? On hydrolysis of sucrose, we are getting glucose plus fructose, right? On hydrolysis of lactose, we are getting glucose plus galactose. On hydrolysis of this maltose, what we are going to get? What will be the monosaccharide we can get on hydrolysis of maltose? Very good. That will be the glucose and glucose. So glucose will be the monosaccharide here. Glucose and galactose will be the hydrolysis product of lactose. Okay, that will be not a maltose. Which enzyme catalyzes the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate in glycolysis? fructose 6-phosphate. Here, one more phosphate group is getting added. So definitely the enzyme has to be end with the kinase. So what will be the name of the enzyme? That will be the phosphofructokinase enzyme. And in glycolysis process, in glycolysis cycle, which will be the rate limiting step? Which step will be the rate limiting step, and what will be the reactant, and what will be the product, and what will be the name of the enzyme? And why it is called as a rate limiting step? Or else just to say which is said to be as the rate limiting step in the process of glycolysis. This step is said to be as the rate limiting step. Why? Because the phosphofructokinase will not be synthesized in any other area. Okay. We can't replace this enzyme once it lost. That is why we are saying this as a rate limiting step. Okay. Now, which of the following is not a function of carbohydrates in the body? Then, okay. Then who is going to maintain the regular body temperature other than this carbohydrate? We have a carbohydrate, protein, and lipid. Which function is there? Which function is to regulate? Very good. That will be the lipid. Okay, this will act as an insulating material for our body. Okay, very good. Adipose tissue. It is stored in the adipose tissue. Fine. In the citric acid cycle, which compound is first formed by the condensation of acetyl-CoA with oxaloacetate? The only thing you have to remember the cycle. That's all about the metabolism. And what will be the name of an enzyme which involves in this particular step? Acetyl-CoA plus oxaloacetic acid is giving the citrate. Okay. And which enzyme is catalyzing this particular reaction? Name of an enzyme. What will be the name of an enzyme which catalyzes this particular reaction? Everybody forgot, right? Kindly go through the cycle once again. Okay. Right. What is the net ATP yield from the complete oxidation of 1 molecules of glucose via glycolysis and the citric acid cycle? On complete oxidation of 1 molecules of glucose in this process. Here, the question will be asking glycolysis and citric acid, not entirely the cellular respiration. Net ATP yield from the complete oxidation of 1 molecules of glucose via glycolysis and citric acid cycle. See here, once again, the same mistake you are doing again and again. Okay, in the question, here I don't ask the net ATP from the entire cellular respiration process. Instead of that, I asked only the ATP, not even an NADPH2 and FADH, net ATP yield in the process of glycolysis and citric acid cycle. Okay. So you have to be very careful when you answer these type of questions, right? Fine. Which enzyme converts glucose 6-phosphate to glucose in liver during glycogenolysis? Here the glycogen is getting breaking down. Okay, I didn't teach this, right? So just skip this question. Glycogenolysis. Okay, fine. Which of the following is not a polysaccharide? Starch, glucose, glycogen, everything will be the polysaccharide, and sucrose will be the disaccharide. Okay. Which of the following enzyme is involved in both glycolysis and citric acid cycle? Quick. See, if you don't know the answer, if you know about all these enzymes, you can easily eliminate the option, right? See here, isocitrate dehydrogenase catalyzes the conversion of isocitrate to alpha ketoglutarate in the citric acid. This is also getting involved in the glycolysis. See here, it will be the answer is aldolase. Okay, don't confuse with this answer. Now, which molecule serves as key regulator of glycolysis by allosterically activating phosphofructokinase? Here, which molecule acts as a key regulator of glycolysis by allosterically activating the phosphofructokinase? See, when you study about the enzyme, you should know what is meant by allosteric enzyme. What is allosteric enzyme? Allosteric enzyme. Anybody know what is meant by allosteric enzyme? See here, this particular enzyme have an additional binding site. Okay. Other than the active site, these enzymes have the additional binding site. That will be the allosteric enzyme. Here, the question asking here will be the which molecule serves as a key regulator of glycolysis by allosterically activating this phosphofructokinase. Now the effector, see now whatever it is getting allosterically attached with this particular fructokinase, that will be calling as the effector. Okay, one will be the active site and one will be the one will be the binding site other than the active site through which the effector can come and bind. The question is what will be the effector? Here, see here, there will be an inhibitor for this PFK and there will be the stimulator for PFK. If ADP is present, some of the enzyme is getting down. If ATP is present, some of the enzyme will getting not activated. Okay, like that. What will be the stimulator here? What will be the effector? That will be the question here. And the answer will be the ADP. Okay, if ADP has to be present, not ATP. Okay. Now that this ADP is an effector, it is act as an allosteric activator of the PFK. This is the one stimulating the glycolysis process whenever the energy levels are low. Right? Now, in the pentose phosphate pathway, which molecule is produced as a precursor for nucleotide synthesis? Okay, I'm sorry. Pentose phosphate pathway is not, I didn't teach that. Okay, fine. Which enzyme catalyzes the conversion of pyruvate to acetyl-CoA in mitochondria? Pyruvate decarboxylase enzyme, isn't it? Sorry. Pyruvate dehydrogenase enzyme. Okay, see, pyruvate decarboxylase. The process of this particular reaction will be the pyruvate decarboxylation. Okay, but here the enzyme will be catalyzing this reaction will be the pyruvate dehydrogenase. enzyme. Okay, don't confuse with the words. See, it is not a carboxylase. As it is will be the decarboxylation reaction. There will be the elimination of carbon dioxide. There will be the production of NADH2. Right? Now. Okay. Answer this question. Which of the following is not a characteristics of enzyme involved in the metabolic pathways? What will be the function of an enzyme? If you know what is enzyme, you can easily scrutinize the option. Here, they are typically specific for their substrate. Here they are saying they alter the equilibrium of a reaction, right? And they are often regulated by the feedback inhibition. They are consumed during the reaction. Odd statements will be there and you have to find out the wrong statement. And they are consumed during the reaction. Is this statement is right or wrong? They are consumed during the reaction. They alter the equilibrium. See, they are consumed during the reaction. The function of an enzyme just to speed up the reaction. Here, see, this will be the enzyme. Whenever it has been bind to the product, there will be the formation of enzyme substrate complex. When it is bind, the substrate is getting converted into the product and the enzyme is getting released from the product. Okay. So here they are not consumed during the reaction because the only point of having this particular enzyme will be to speed up the reaction. Okay. Now just remember that. Now, which of the following statement about glycogen? Okay. Relax. Next, pause. Okay. During glycolysis, which compound is converted into phosphoenol pyruvate by the enzyme enolase? See, something is getting converted into PEP by an enzyme called the enolase. What will be the reactant here? This is what they are asking. Okay. Just whenever they are asking like question like this, just simplify the question. Yeah, in which compound is converted into PEP? Something is getting converted into PEP by an enzyme called enolase, and what will be the reactant here? Whether that will be the 3-phosphoglycerate, 2-phosphoglycerate, that will be the 3-phosphoglycerate. See, the enolase enzyme catalyzes the 2-phosphoglycerate. Okay, that will be the answer here. Now, see, just remember the cycle. Okay, then whatever the question they asked in the examination, you can easily answer there if you know the question. And in tomorrow's class, I am going to explain the glycogenolysis, gluconeogenesis, everything. Okay. Fine. That's all for today. And for tomorrow, the remaining topic in the physiology will be completed. Soon in tomorrow's class. Okay. Is there any doubts regarding today's class or any other thing? A pentose phosphate pathway, gluconeogenesis, mitochondrial electron transport chain, and oxidative phosphorylation. This thing will be the pending, and reproduction in mammals will be the pending. Okay, that's all about the physiology. And tomorrow everything will be completed. Okay. And if you have any doubts, you can ask me. Just 1 more minute is there, or else you just leave the class.